Welcome to our deep dive today. We are going to be talking about beta-haemolytic Streptococci. You know, those tiny troublemakers that we find all the time in the clinical setting? We'll be going through Principles and Practice of Clinical Bacteriology. It's the second edition by Gillespie and Hockey. We are going to be focusing on the stuff that's most important for medical microbiologists.
Yeah, it's pretty amazing. We classify these organisms by how they do hemolysis, but they have such a huge range of different virulence factors and ways that they present.
Yeah, for sure. It's more than just, looking at blood agar and seeing clear zones. So let's kind of start with the basics. So we're talking about gram-positive Cocci in chains. And they do complete hemolysis, right? That's the key thing. They completely break down the blood on blood agar plates. But how do we tell all of these different beta-haemolytic streps, apart from each other?
Well, that's where Lancefield Grouping comes in, right? It's what we've used forever. We use it to identify strep. We can figure out which group they are. Like, are they group A, B, C, G, and so on. Because we're targeting these really specific carbohydrate antigens. And it's really helped us a ton in figuring out how these things spread and how important they are clinically.
Yeah, it's like we're putting them into these different families. And each family has its own quirks. So beyond just putting them into those groups, what about their ability to cause disease? What really makes them different from each other?
So it all really comes down to virulence factors. It's complicated, but basically it's both stuff that's actually part of the cell wall and stuff that the bacteria release. And here's the thing, even within one species, like say Streptococcus pyogenes, GAS, there's just a ton of variation in those factors from strain to strain.
So it's like every strain has its own little bag of tricks. For GAS, though, M protein is a big deal, right? It's involved in immune evasion, but also in how it sticks to our cells. And even in some of those weird autoimmune things that can happen afterwards.
Absolutely. The M protein is like the key thing that makes GAS virulent. It can bind to this protein we have, factor H. Factor H controls our complement system. So basically, it lets GAS get around our innate immune response. And there are tons of different M protein types. We found over 100. So that helps explain why we see so many different things clinically with GAS.
Wow, so it's like it has all these disguises, always changing, so our immune system can't keep up.
No.
But it's not all about hiding, is it? GAS also pumps out all these toxins, and they can really do some damage, like those hemolysins.
Yeah.
Streptolysin O and streptolysin S, those are what cause those clear zones on blood agar, right?
Exactly. They can really wreck a lot of different cells in our body. Streptolysin O is interesting. It doesn't work so well when there's oxygen around, so it's more active in tissues where there's not a lot of oxygen. And we can actually use it in blood tests to see if someone has had a GAS infection before.
Oh, wow. So it's not just something we see on a plate. It can tell us something about a patient's history. What about streptokinase? That one seems pretty sneaky.
Right.
It helps GAS spread because it breaks down blood clots.
It is pretty clever. It uses our own system against us. Basically, it activates something called plasminogen, and that's what usually breaks down clots. But GAS uses it to move around more easily.
It's like GAS is hijacking our bodies. But the worst thing GAS has got to be superantigens, right? They cause those massive immune reactions. And sometimes even toxic shock syndrome.
Superantigens can be scary. They really mess with our immune system. It's different from a normal antigen. Normal antigens activate just a few T cells. Superantigens turn on tons of T cells, so you get this flood of cytokines. That's what leads to the problems with toxic shock syndrome.
It's like hitting the emergency button on our immune system, just all this inflammation going crazy.
Yeah.
And then potentially organ failure. And GAS has a bunch of these different superantigens. I remember reading about SPEB. It's an enzyme, a cysteine protease. I think they thought it was a superantigen at first. What does it do?
Yeah, SPEB is interesting. We are always learning more about how these virulence factors work. So it's not a superantigen, but it's a powerful protease. It chops up all kinds of proteins we have, like immunoglobulins and complement components. So it messes with our immune system.
So it's breaking down our defenses, making it easier for GAs to spread. What about SIC, the streptococcal inhibitor of complement? That one seems to be important in those M1 strains that cause those really bad invasive infections.
Yeah, SIG is another piece of the puzzle with GAS. It binds to the membrane attack complex. That's the last step in the complement cascade. So it stops those cells from being lysed by complement. And what's crazy is how fast it changes in the body. Like it's always trying to stay one step ahead of our immune system.
Like a constant arms race between GAS and us. So we've talked about those things that are part of GAS and the toxins it releases. But GAS also has ways to stick to our tissues. And it can even get inside cells, right?
Yeah, definitely. Sticking to our cells is the first step for it to cause an infection. And GAS uses a bunch of different things to grab on. One is called lipoteichoic acid, or LTA. It binds to fibronectin on our cells.
Oh, so it's like the first point of contact.
Yeah, a good way to put it. And then other things come in and make that attachment stronger so it can really hang on, like the M protein and those fibronectin binding proteins. They basically act like grappling hooks, latching onto our cells and letting GAS hang on.
So not just evading, but actually like attacking.
Yeah.
What I find really interesting is, we always think of GAS as an extracellular pathogen, but it can actually get inside some of our cells, right?
That's right. It's not how it usually survives. But studies show it can get into epithelial cells and even hang out inside them.
Huh. I didn't realize that. Why would it go to all that trouble? I mean, it can cause so much damage just being outside of the cell.
That's a good question. We think maybe it's a way to hide from the immune system, avoid those immune cells. And maybe that's how it causes those infections that just never seem to go away.
Like a little hide-and-seek game. So all of this craziness with virulence factors, it's not just random, is it? There's all these complex systems that control them.
Absolutely. GAS has evolved all these ways to change its virulence, depending on the environment.
So it's like it knows when to ramp things up and when to lay low.
Yeah, kind of like a conductor, turning genes on and off when it needs them.
One of the big ones is MUGA, right? Multiple gene activator. It can sense things like how much carbon dioxide there is, or if there's enough iron around.
That's one of the major players. It's a two-component system. It senses those cues you mentioned, and then it goes and controls a whole bunch of genes related to virulence.
So it's like a master switch.
Exactly.
And then there's that CSRS system, or co-VRS. I think I've heard it called both ways. It's more like a fine-tuning knob.
Right. It controls things like the capsule.
Yeah.
And streptokinase. And some of the toxins we talked about, like SLO and SPEB.
Mm-hmm. So it's all about striking the right balance. Cause enough damage, but don't get caught. It's just amazing how much we know about these systems now. It really shows how complicated GAS is.
Yeah, it's not simple at all. And we can use this information to try and find new treatments, things that might disrupt those control systems, instead of just relying on antibiotics.
That's really cool. Find that weak spot and exploit it.
Exactly.
And speaking of new approaches, genome sequencing has really changed how we think about GAS. And all the other beta-haemolytic strep, too. Now we have these detailed maps of their genes.
Oh, yeah. It's been huge. It's like having the instruction manual. We can see all the virulence factors and how they've evolved.
Those first GAS genomes we got, they really showed how diverse those virulence genes are.
Yeah.
But they also showed us something else, right? Those mobile genetic elements, like bacteriophages.
Right. So not just the genes themselves, but these little chunks of DNA that can jump around between strains or even between species. They can move virulence genes around and create new combinations.
So it's like they're swapping weapons.
Yeah.
And sometimes they make even deadlier ones.
And since then, we've sequenced a lot more genomes. And we've seen that those phages and other elements are really important for how GAS changes. They're driving those outbreaks we see, where there's a lot of really severe invasive disease.
Like those M3 isolates. I remember reading about those.
Yes.
They caused a big jump in invasive GS cases. And it turns out they had picked up some particularly nasty superantigens.
Yeah. Those new M3 isolates have a version of the superantigen SPE. It's called SPE3, and it's way more powerful than the old one, SPEA1. They've also picked up a phage that carries two other virulence factors. One's another superantigen, SPIC, and the other is an enzyme that breaks down lipids, SLO.
So they've basically supercharged their arsenal. Makes you realize these bacteria are always evolving.
Yeah.
And we have to keep up.
Absolutely. And it's not just about GAS. We have the complete genome sequences for a couple of GBS strains now, too. One's serotype 3 and the other is serotype V. And they've given us some great clues about how GBS causes disease.
And have we seen the same kind of thing with those GBS genomes, those mobile elements and maybe new virulence factors?
Oh, yeah, definitely. Those GBS genomes have tons of potential virulence genes that are linked to those mobile elements. So horizontal gene transfer is definitely happening in GBS, too.
So they're all sharing genetic information and potentially creating new threats.
Exactly. It's why we need to keep doing genomic surveillance to track these changes and hopefully catch those new threats early on.
So we've been talking about all the crazy things these bacteria can do, but let's take a step back and talk about how we identify them in the lab. It all starts with those basic microbiology techniques, right?
Absolutely. Those tried and true methods are still essential. They're the foundation of how we diagnose these infections.
So we're talking colony morphology, gram stain, catalase test, and of course Lancefield grouping.
Exactly. You always start by looking at those colonies on blood agar. You want to see those zones of beta-haemolysis. That's the first clue.
Like their calling card.
Right. And then you do a gram stain. You're looking for those gram-positive Cocci in chains, and the catalase test is important to make sure it's not staph. Staph are also gram-positive Cocci, but they're catalase positive, unlike strep.
So a simple test, but it helps narrow things down. And then we have Lancefield grouping, the gold standard for beta-haemolytic strep.
Yeah, Lancefield grouping is still key. It's based on those specific carbohydrate antigens, and it lets us classify those bacteria really quickly and accurately into those groups we all know, A, B, C, G, and so on.
So we can put them into their little families, each with their own personality.
Exactly. And that helps us decide on treatment and maybe anticipate any complications. But sometimes Lancefield grouping isn't enough.
When would we need to go further?
Sometimes those initial tests don't give us a clear answer. Or maybe we think it might be a rarer species, so then we have to look at their biochemical profile. We use those commercial identification systems. They give us a more complete picture of what those bacteria can do, help us pin down exactly what species it is.
So it's like our detective kit. But in the clinical world, sometimes we need answers fast, especially with something like sepsis or meningitis. Absolutely. Waiting for those cultures and biochemical tests can take too long.
It's like forever when you need to act quickly.
Exactly.
Yeah.
That's where those rapid identification assays come in. We can detect the antigens directly, without waiting for cultures.
Those rapid tests have been amazing, especially for GAS and GBS.
Yeah. For GS pharyngitis, we use rapid antigen tests all the time. They target either the Group A carbohydrate or the M protein.
So much faster than waiting for a culture. It means doctors can start treatment right away and hopefully prevent those bad things from happening, like rheumatic fever.
Exactly. But we have to remember that these tests aren't perfect. They're really specific, but sometimes they miss cases.
So you can get a false negative, especially if there's not a lot of bacteria there.
Yeah. So doctors often use those rapid tests, along with the throat culture, just to be sure.
So the right tools for the job.
Right.
For GBS. Those rapid tests are mostly used to find women who are carriers when they're in labor. Right. It's about preventing those really bad infections in newborns. Early onset GBS disease.
Because it usually comes from the mother's bacteria.
Exactly. If we know the mother is a carrier, we can give her antibiotics during labor, and that really cuts down the risk of passing it to the baby.
So it's all about prevention.
Right.
We usually use cultures for prenatal screening.
Right. That's still the gold standard to see if a woman is carrying GBS. But those rapid tests are useful too, especially when you need an answer quickly.
So faster results, but maybe not quite as accurate.
Yeah, that's the trade-off. They're not as good as cultures in terms of sensitivity and specificity.
So doctors have to take that into account.
Absolutely. But scientists are working on new rapid tests to try to make them even better so we can get those fast results without sacrificing accuracy.
So besides just figuring out if it's strep or not, sometimes we have to do typing to figure out those patterns of how it spreads and if there might be an outbreak brewing.
Right. Typing is important for understanding those different strains.
So it's like we're giving them fingerprints so we can track them.
Yeah, a good way to put it. And for GAS, the classic way to type them is using those T and M proteins. It was developed by Griffith and Lancefield.
So it's a serological typing system, using antibodies to find those specific proteins on the bacteria.
Exactly. M typing focuses on the beginning of the M protein, the N-terminal sequence, because that part is different between strains.
So each M type means there's a different version of that protein.
Right. There are over 100 different M types. That's how diverse this species is. And to make things even more complicated, each M type can also have different T antigens. Those are based on other cell wall proteins.
So it's like a two-part code.
Exactly. And some GAF strains also produce this thing called serum opacity factor, or OF. We can use that for typing too. But these days, we're moving away from those traditional serological methods, and we're using molecular techniques more and more.
Molecular typing is really changing things.
Yeah, it's much more powerful.
And for GAS, the main method is M gene sequencing.
Right. We sequence a part of the M gene. That's the gene that codes for the M protein. So it's like a molecular version of M typing. And we can identify all those different M types.
More precise than the old way.
Definitely. We've already found over 150 amine types, more than the traditional M types. And we can group those M types into subfamilies based on how similar their sequences are and how they're arranged on the chromosome.
So we can build those family trees.
Yeah.
And see how all those GA strains are related.
Exactly. And this is really important for epidemiology. We can track how specific strains spread. and it helps us understand how to make better vaccines. We also use other molecular methods for GAS like PFGE and MLST.
PFGE, that's the one with the DNA fingerprints, right? And MLST looks at several different genes.
Yeah. PFGE stands for pulsed field gel electrophoresis. It separates large pieces of DNA so you can see those differences between strains. MLST is multi-locus sequence typing. It analyzes the sequences of several housekeeping genes.
So they both give us info about the strains, but they have their pros and cons.
Right. PFGE is really good at distinguishing between strains, but it takes more work. MLST is more standardized, so it's easier to compare results between labs. And there are those PCR-based methods too, like RAPT and PCR-RFLP. They're good for quick screening.
So we've got all these different tools, and we pick the right one for the job.
Exactly.
Now, what about GCS and GGS? Those are the ones that cause those infections that are kind of like GAS, but usually not as bad. Have their typing methods changed too?
They definitely have. We used to do things like bacteriophage typing and bacteriocins, but now we mostly use serological and molecular methods, just like with GAS.
So what are the main typing schemes for them?
Just like GAS, we have a serological system based on T proteins. We found over 20 different T protein types in GCS and GGS that infect humans.
So even though they're not as well known as GAS, they're still pretty diverse.
Absolutely. And we can also use M proteins for typing. And just like in GAS, those M proteins are linked to how virulent the strain is. But we don't use these serological schemes as much as for GAS. They're mostly done in specialized labs.
Makes sense. They're not as common as GAS. What about molecular methods?
We use those too. PFGE and ribotyping have been really helpful, especially for figuring out outbreaks.
Ribotyping looks at ribosomal RNA genes, right? It's like another way to get those fingerprints.
Exactly. It gives us a stable marker for comparing different strains. And we've even started using M gene sequencing for GCS and GGS, just like we do for GAS. And we're finding all sorts of different M types in those species too.
So they're changing and adapting all the time.
That's right. But unlike GAS, we don't know as much about those GCS and GGS M types yet, like which ones are more likely to cause disease.
So there's still a lot to learn.
Definitely. But those molecular tools are helping us unlock those secrets.
And what about typing GBS? I know we've always used serotyping for that, but have we started using molecular methods for GBS too?
Yeah. For GBS, serotyping has been the go-to method.
Yeah.
It's based on those capsular polysaccharide antigens. We've identified 9 different serotypes, from IA to AF.
So each serotype means a different version of that capsule.
Exactly. And those serotypes are important because some are more likely to cause certain diseases, and they also guide vaccine development.
So it's useful for both treating patients and tracking outbreaks.
Absolutely. But serotyping has its limits. It can't always tell us the difference between closely related strains or strains that have picked up new capsule genes through horizontal gene transfer.
So it's a good starting point, but we need more.
Right. And that's where those molecular methods come in. PFGE and ribotyping have been really useful for GBS. They can show us those subtle differences between strains that serotyping might miss.
Those techniques are so versatile.
Definitely. But one technique that's been particularly helpful for GBS is MLEE, multi-locus enzyme electrophoresis.
MLE, that's the one that looks at enzymes, right? Like tiny differences in those enzymes.
Exactly. It analyzes variations in metabolic enzymes, little changes in the amino acid sequence. That can change how they move in an electric field. By comparing those enzyme patterns, we can figure out how closely related different isolates are. But MLEE has some problems, especially with those serotype 3 strains. Those are the ones that often cause those really serious infections in babies.
So it's not always the best choice for GBS.
Right. That's where MLST has become really important.
MLST, the one that analyzes DNA sequences.
Exactly. MLST stands for multi-locus sequence typing. We sequence those little bits of 7 different housekeeping genes, and we use that to classify those isolates into sequence types, or STs.
So each ST is a unique combination of those gene variants?
Exactly. It's a really robust and standardized way to compare strains. And what's really interesting is we found some links between specific STs and the types of diseases they cause.
Oh, that's fascinating. Like what?
Well, for example, ST1 and ST19 are mostly found in people who are just carriers. They don't have symptoms. But ST23 can be found in carriers and in people who are actually sick.
So some are more likely to cause problems than others.
Exactly. And then there's ST17. That one's really nasty. It's almost always found in babies with those serious invasive infections.
Wow. So MLST can tell us a lot about those risks.
It definitely can. It's helped us understand a lot about GBS epidemiology and virulence. And it's also helped us understand this thing called capsular switching, where GDS strains can actually change their serotype by picking up capsule genes from other strains.
So they're trading genes and maybe becoming more dangerous.
Exactly. It just shows how dynamic these genomes are and why we need to keep watching them closely.
So we've got all these different typing methods, but how do we make sure they're accurate and that everyone's getting the same results? I mean, if we want to track these things properly, we all need to be on the same page, right?
Absolutely. Standardization is really important. That's where quality assurance programs come in.
Yeah, like some kind of global quality control for strep typing.
That's a good way to think about it. Basically, these programs send out strains that we already know a lot about to different labs, and then everyone does their typing, and we compare the results. So if there are any differences, we can figure out why.
Like a big test to make sure everyone's doing things right.
Exactly. One of the first programs for GAs typing started back in the late 90s. It had 6 big strep labs from around the world.
Wow, that's awesome. Really shows how scientists can work together.
Definitely. They sent out GA strains a few different times, and everyone did their typing, both traditional methods and the newer molecular ones. And the results were really good. Most of the labs got the same answers.
That's good to hear. But did they find any problems?
There were a few times where the results didn't match up, and that helped us realize we needed to be really clear about how we define those different GAS types. So everyone's using the same criteria. It's a work in progress. We're always trying to improve those methods.
Always getting better.
And now those programs have grown. There are even more labs involved, so we have even better global surveillance.
It's amazing how much we can learn when we all work together. Now let's talk about antibiotic resistance. That's a big concern for everyone. But those beta-haemolytic strep have usually been pretty sensitive to penicillin, right?
Yeah, they have. Penicillin's been our go-to drug for these infections for a long time, and thankfully they're still mostly susceptible to it.
That is good news. But we can't get complacent, right? We have to keep watching those resistance patterns.
Absolutely. Resistance is always changing. Bacteria are always finding ways to beat our drugs. Penicillin resistance is still pretty rare in those streps. But we are seeing some worrying trends with macrolides.
Yeah, those are important because we use them when people are allergic to penicillin. So if resistance keeps going up, it'll be a big problem.
Definitely. Doctors need to know those local resistance patterns and consider other options if macrolide resistance is common in their area.
So it's about choosing the right drug for the situation.
Exactly.
What's causing this increase in macrolide resistance?
There are 2 main ways it happens. One is called target site modification. The bacteria change the part of the ribosome where the antibiotic binds, so the drug can't get in and stop protein synthesis.
So it's like they're changing the locks on their doors.
Exactly. And these changes are usually made by enzymes called methylases. They add a methyl group to the ribosome, and that blocks those macrolides.
And those methylases come from genes, right? Like genes. And those genes can spread between bacteria.
Right. Those genes, especially B and TR, are found in a lot of GAS and other strep, and they're definitely contributing to that macrolide resistance.
It's not just one strain evolving, it's those genes hopping around.
Exactly. Makes it a lot harder to fight.
What about that other mechanism, active drug efflux?
That's another sneaky way those bacteria can get around antibiotics. They basically make these pumps, called efflux pumps, that sit in their cell membrane.
So they're kicking those drugs right back out.
Exactly. The pump grabs those antibiotics and throws them out of the cell before they can do any damage.
And of course, there are genes for those pumps too.
Right, like the MEF-A gene. It's common in GAS and other streps, and sometimes those bacteria have both and MEF-A genes, so they're resistant to multiple drugs. That makes them much harder to treat.
It's like they're always one step ahead.
It's a constant challenge.
So how do we treat these infections? Penicillin's still the first choice, right?
Yes, it is. Penicillin is still our gold standard.
Yeah.
And it works really well for most beta-haemolytic strep.
So that old drug is still the best.
For simple infections, like strep throat, we usually give penicillin V pills for about 10 days.
It's important to finish all those pills.
Right. Even if you feel better, you have to take the full course to make sure all the bacteria are gone and to prevent complications.
And for those people who can't take penicillin?
We usually give them macrolides, like erythromycin or azithromycin. But with resistance going up, it's really important to check those local patterns before prescribing those.
So you have to weigh the risks and benefits.
Exactly. And If resistance is a problem, there are other options, like clindamycin or some of those older cephalosporins.
Knowing your local antibiogram is really important.
Absolutely. For those more serious infections, like invasive GAS disease or GBS sepsis, we need to be more aggressive. We usually give penicillin G intravenously.
So you get those high doses right into the bloodstream.
Exactly. And in really severe cases, especially those with toxic shock syndrome, we often add clindamycin to the penicillin.
Why clindamycin?
Well, clindamycin stops protein synthesis, but it also does something else. It blocks the production of those toxins, including those superantigens.
So it hits those bacteria in two different ways.
Exactly. And studies have shown it really helps in those really sick patients.
Sometimes, though, antibiotics aren't enough. Like if the bacteria have gotten into deep tissues.
You're right. In cases of necrotizing fasciitis, for instance, you have to do surgery to remove all the dead and infected tissue. That's the only way to really control the infection.
So it takes a whole team to deal with those really bad cases.
Absolutely.
What about other things we can do besides antibiotics?
There are a few things. One is intravenous immunoglobulin, or IVG. We sometimes use it in those severe cases, especially those with toxic shock syndrome.
IVG, that's like a concentrated dose of antibodies, right?
Exactly. It's purified antibodies from healthy donors. We use it for all sorts of immune problems. We're not exactly sure how it works in strep infections, but we think it might neutralize those toxins, help with killing the bacteria, and calm down the inflammation.
Like giving the immune system a helping hand.
Right. We don't have a ton of hard evidence for IVE in strep. Most of the studies are just observations, but it seems like it could be helpful.
More research is always a good thing.
Definitely.
What about those phage therapies? I've heard more and more about those.
Yeah, phage therapy is getting a lot of attention these days, especially with antibiotic resistance being such a big problem. Phages are viruses that attack bacteria, and they're very specific. They only kill certain types of bacteria, and they can multiply inside the bacteria, so you get this amplified effect.
So it's like a targeted strike.
Exactly. There are still some things to work out with phage therapy, like how to prevent resistance to the phages and getting those regulatory approvals. But it has a lot of potential, especially for those infections that just don't respond to antibiotics.
An exciting area for sure.
Absolutely.
What about vaccines? Vaccines have been so successful for other diseases. Any chance we'll have a vaccine for those beta-haemolytic strep?
A vaccine would be amazing, especially for those people who are most at risk, like babies and those with weakened immune systems.
What's the latest on a GAS vaccine?
Most GAS vaccine research has focused on the M protein because it's the main target for protective antibodies. But the problem is those M types, there's so many different ones, over 100.
It was hard to make a vaccine that works against all of them.
Exactly. The first GAS vaccines only targeted a few M types, so they didn't work that well. But now we're working on multivalent vaccines. They include several different M types.
It's like a multi-tool.
Right. Those early trials of these multivalent vaccines look good. People are making antibodies against a bunch of different GAS serotypes, but we're still worried about those potential autoimmune problems, where those antibodies might attack our own tissues.
Right, because of those similarities between bacterial and human proteins.
Exactly. So scientists are being very careful. They want to make sure any new vaccine is both safe and effective.
And what about a GBS vaccine?
There are some promising GBS vaccines in development, too. They focus on those capsular polysaccharides. Those are the main things that make GBS so dangerous in babies.
Those early GBS vaccines targeted serotype 3.
Right, that's the one we see most often in babies with those bad infections. But we know those strains can switch their capsules. So a vaccine against just one serotype might not be enough. That's why we're developing polyvalent GBS vaccines that go after multiple serotypes.
So like those GAS vaccines, they're going for broader coverage.
Exactly. And they're often conjugated to proteins to make them work even better. The results from those early trials have been encouraging.
So maybe we'll have vaccines for both GAS and GBS soon.
It's definitely possible. And those vaccines could really reduce the number of people getting sick, especially those who are most vulnerable.
It would be a huge step forward.
Absolutely. We need to keep pushing those research efforts to make those vaccines even better and make sure they're safe and effective so we can finally protect people from these infections.
Well, this has been an amazing look at those beta-haemolytic strep.
It has been fascinating.
We've covered a lot of ground. From those basic features to all those complex things they do to cause disease, from how we diagnose them to how we treat them. and even those new therapies and vaccines that are in development.
It's been a pleasure talking about these bacteria with you. I hope everyone listening feels like they've learned something new.
I think our listeners are ready to take on any strep challenge now.
Ah,
But before we go, let's do a little quiz, just 5 questions, to see how much you remember.
I'm game.
Okay, first question. How does that M protein on GAS help it avoid our immune system?
Okay, so the M protein binds to this thing called factor H. It's one of those complement control proteins. So basically, it stops those complement cascade from activating, and that's how GAS avoids being destroyed.
Perfect. Second question. What's the big difference between those early onset and those late onset GBS infections in babies?
Early onset GBS usually shows up in the first week of life. The baby gets from the mom during birth. Late onset GBS happens after that first week, and it can come from different places, like the hospital.
That's right. Okay, third question. Which molecular typing method is used all the time for GAS? The one that has found over 150 different M types.
That would be M gene sequencing. We look at the DNA sequence of that M gene. That makes us M protein.
You got it. Question number 4. How does clindamycin work? And why do we like to use it for those really bad strep infections, especially those that have toxic shock syndrome?
Clindamycin blocks protein synthesis in the bacteria, but it also stops them from making those toxins, like those superantigens, that are the main culprits in toxic shock syndrome.
Exactly. And last question. What's one of the biggest hurdles in making a good GAS vaccine, given how diverse they are?
The biggest problem is those M protein types. There are just so many of them. It's hard to make a vaccine that protects against all those different strains. That's why scientists are working on those multivalent vaccines that include several different M types to try and get broader coverage.
That's right. You aced the quiz. Looks like you learned a lot today.
I did. It's been a great review.
Well, that wraps up our deep dive into those beta-haemolytic strep. Until next time, keep learning and stay curious about those amazing microbes all around us.
And don't forget to wash your hands.